In this video you will be able to know everything about antibodies, and the explanation will contain the following points in detail. What are antibodies and how they are working in our bodies? The antibody structure and classes. The 5 main classes of antibodies: IgG, IgM, IgE, IgA and IgD. The normal reference values of the antibodies classes. What if you have low levels of antibodies, causes and symptoms? What if you have high levels of antibodies, causes and symptoms? The best foods for boosting your immune system. Antibodies in medicine and research. What are antibodies? Antibody, also called immunoglobulin, a protective protein produced by the immune system in response to the presence of a foreign substance called an antigen. Antibodies recognize and latch onto antigens in order to remove them from the body. A wide range of substances are regarded by the body as antigens, including disease-causing organisms and toxic materials such as insect venom. How antibodies work. When an alien substance enters the body, the immune system is able to recognize it as foreign because molecules on the surface of the antigen differ from those found in the body. To eliminate the invader, the immune system calls on a number of mechanisms, including one of the most important, antibody production. Antibodies are produced by specialized white blood cells called B lymphocytes or B cells. When an antigen binds to the B cell surface, it stimulates the B cell to divide and mature into a group of identical cells called a clone. The mature B cells, called plasma cells, secrete millions of antibodies into the bloodstream and lymphatic system. Phagocytic cells destroy viral and bacterial antigens by eating them, while B cells produce antibodies that bind to and inactivate antigens. Encyclopaedia Britannica, Inc. Antibodies and B cells. B cells and antibodies together provide one of the most important functions of immunity, which is to recognize an invading antigen and to produce a tremendous number of protective proteins that scour the body to remove all traces of that antigen. Collectively, B cells recognize an almost limitless number of antigens. However, individually each B cell can bind to only one type of antigen. B cells distinguish antigens through proteins, called antigen receptors, found on their surfaces. An antigen receptor is basically an antibody protein that is not secreted but is anchored to the B cell membrane. All antigen receptors found on a particular B cell are identical, but receptors located on other B cells differ. Although their general structure is similar, the variation lies in the area that interacts with the antigen, the antigen binding, or antibody combining, site. This structural variation among antigen binding sites allows different B cells to recognize different antigens. The antigen receptor does not actually recognize the entire antigen. Instead, it binds to only a portion of the antigen's surface, an area called the antigenic determinant or epitope. Binding between the receptor and epitope occurs only if their structures are complementary. If they are, epitope and receptor fit together like two pieces of a puzzle. An event that is necessary to activate B cell production of antibodies. Antibody structure and classes. Each antibody molecule is essentially identical to the antigen receptor of the B cell that produced it. The basic structure of these proteins consists of 2 pairs of polypeptide chains, lengths of amino acids linked by peptide bonds, that form a flexible Y shape. The stem of the Y consists of one end of each of 2 identical heavy chains, while each arm is composed of the remaining portion of a heavy chain plus a smaller protein called the light chain. The two light chains also are identical. Within particular classes of antibodies, the stem and the bottom of the arms are fairly similar and thus are called the constant region. The tips of the arms, however, are highly variable in sequence. It is these tips that bind antigen. Thus each antibody has two identical antigen binding sites, one at the end of each arm. And the antigen binding sites vary greatly among antibodies. This image demonstrates the variable and constant domains of an antibody. Antibodies are grouped into 5 classes according to their constant region. Each class is designated by a letter attached to an abbreviation of the word immunoglobulin, IgG, IgM, IgA, IgD, and IgE. The classes of antibody differ not only in their constant region but also in activity. For example, IgG, the most common antibody, is present mostly in the blood and tissue fluids, while IgA is found in the mucous membranes lining the respiratory and gastrointestinal tracts. The 5 main classes of antibodies. 1. Antibody, IgG. These bodies defend the body against bacteria and viruses, and are found in various body fluids, as they are the smallest in size, and therefore move easily in cells. 2. Antibody, IgM. They are the largest among antibodies, and are found in mucus, tears, saliva, and blood, as well as some other areas of the body. These bodies are produced in response to a viral or bacterial infection, and they prevent enemies from infecting cells with damage, as well as preventing them from reaching the internal organs. 3. Antibodies, IgE. These bodies react to allergens, whether dirt and dust, fungi, or food allergens, as well as protect the body from parasitic worms. These bodies are found in the skin, lungs and mucous membranes, and are found in a small percentage in the blood. 4. Antibody, IgA. These bodies are found in a large proportion in the mucous membranes, especially in the lining of the respiratory and digestive tracts, and are also present in tears and saliva. These bodies work to prevent the entry of foreign and harmful bodies into the blood circulation. 5. Antibody, IgD. It is the lowest type in the body, is found in small quantities in the blood, and is usually present with IgM antibodies, and it enhances the activity of immune cells. Immunoglobulin's blood test is carried out to measure the level of types of antibodies in the blood. The immune system makes antibodies to protect the body from bacteria, viruses, and allergens. Immunoglobulin normal reference values. These values are for adults older than 18 years. IgG 639 to 1349 mg per deciliter. IgM 56 to 352 mg per deciliter. IgA 70 to 312 mg per deciliter. IgE 1.53 to 114 mg per deciliter. IgD 1.30 to 152 mg per liter. What if you have low levels of immunoglobulins? This case is called hypogammaglobulinemia. That means your immune system isn't working as well as it should be. This may be caused by 1. Infection with some diseases like allergies, chronic infections, an autoimmune disorder that makes your immune system overreact, such as rheumatoid arthritis, lupus, or celiac disease. liver disease, inflammatory bowel disease, multiple myeloma, lymphoma, or leukemia. AIDS. 2. Chronic stress. Mental health directly affects the functioning of your immune system. So if you suffer from a problem of chronic stress, you are more susceptible to various diseases. According to a report published by the American Psychological Association, chronic stress can weaken the immune system, because it reduces white blood cells that fight pathogens, making you more susceptible to infection with viruses in particular. 3. Sleep disturbances. Sleep disturbances are one of the most important causes of immunodeficiency. If you do not get enough hours of sleep or good quality sleep, you may be more susceptible to various diseases. Lack of sleep and its quality can affect the speed of your recovery from the disease. Because the immune system works to release certain proteins known as cytokines, which your body needs in the event of an infection or inflammation. In addition, lack of sleep can affect the numbers of antibodies and immune cells that are important to protect your body from disease. 4. Personal hygiene. Especially washing hands frequently using soap and water. Hands are a primary carrier of various viruses and disease pathogens, and if you do not wash and sanitize them frequently, you are more likely to contract the disease. 5. Smoking. If you smoke, you are more likely to get many different diseases, by suppressing the work of the immune system. And it may reduce the effectiveness of some treatments. 6. Taking some types of medication. There are some types of medicines that are associated with unwanted side effects, including weak work and efficiency of the immune system, especially cortisone. In addition, undergoing some treatments such as chemotherapy may contribute to immunosuppression. Symptoms of immunoglobulins deficiency. Hypogammaglobulinemia. 1. Fatigue. Feeling tired and stressed is one of the main symptoms of weak immunity, which is a common thing that can occur as a result of stress and lack of sleep, but if you continue to feel tired and stressed, this can be an indication that the immune system is not functioning normally. The level of cortisol plays an important role in regulating the immune system. As high or low cortisol has a negative effect on immunity, leading to a feeling of fatigue. 2. Frequent infections. Recurrent infections are one of the most common symptoms of weak immunity, which means that the immune system is not working well and is unable to fight infection. Inflammation can occur in any part of the body, such as respiratory infection, arthritis, dermatitis, and others, which affects general health and leads to pain in various parts of the body. 3. Cold and sore throat. If you frequently get colds and sore throats, this is likely the result of a weakened immune system and its ability to fight infection. Thus, a person who develops persistent respiratory infections often has a weakened immune system. 4. Allergy. Allergies occur when the immune system reacts abnormally to harmless substances such as dust, mold, or pollen. The immune system may overreact by producing antibodies that may attack allergens, and if you suffer from wheezing, itching, runny nose or watery eyes, this means that the immune system has disorders because it overreacts to allergens. 5. Slow recovery from injury. If the immune system is weak, bacteria can multiply on wounds and injuries because it will not be able to resist them, and thus the recovery period from these infections increases. If the immune system is healthy, this means that the body is able to recover from the infection quickly, as the immune system fights bacteria and prevents their reproduction on the skin. 6. Digestive problems. The health of the gut bacteria is closely related to the health of the immune system. When the gut bacteria are in balance, this means that the immune system is healthy, and vice versa. Food allergies are a major sign and cause of a malfunction in the immune system, and if you suffer from digestive problems such as diarrhea, stomach infections and nausea frequently, this is a sign of a weak immune system. 7. Anemia. Anemia is a condition in which there is a decrease in the total red blood cells or hemoglobin, or a decrease in the ability of blood cells to carry oxygen. And this can occur when the immune system is weak. A weakened immune system treats healthy red blood cells as foreign bodies and attacks and destroys them, which can lead to anemia. Symptoms include tiredness, weakness, shortness of breath, or a decreased ability to exercise. 8. Hair loss. Hair loss is a condition that usually affects the scalp. It can occur as a result of an autoimmune disorder, in which the immune system is overly fluid and attacks the hair follicles, resulting in hair loss. Alopecia areata is a form of excessive hair loss, and indicates a severe immune defect, which can lead to the loss of eyelashes and eyebrows, as well as the presence of noticeable voids in the hair. 9. Joint pain. Joint pain can be caused by an imbalance of the immune system. This causes arthritis because the immune system cannot fight the infection that causes inflammation. A weakened immune system can also lead to autoimmune diseases such as rheumatoid arthritis, where the immune system attacks healthy tissues, resulting in permanent damage to the good cells in the body. Symptoms include joint swelling, redness, warmth, stiffness, pain, and fever. What if you have high levels of immunoglobulins? Reasons for a high level of antibodies in the body. This can be caused by 1. Infection with some diseases like an autoimmune disorder that makes your immune system overreact, such as rheumatoid arthritis, lupus, or celiac disease. Chronic infections, allergies, liver disease, inflammatory bowel disease, multiple myeloma, lymphoma, or leukemia. 2. Exposure to dust. If the body is allergic to dust and dirt, it will cause an increase in the levels of antibodies in the body, and thus its negative effects will appear. 3. Changes in the weather. The weather is one of the main factors that can cause a high level of antibodies in the body, as the chances of negative symptoms increase between seasons, as well as during times when the humidity increases to cause the growth of fungi. 4. Pet handling. Pet fur and saliva can cause allergies, which raises certain antibodies in the body. In the case of pets in the house, precautions must be taken to keep them clean and keep them away from children as much as possible. 5. Eat allergenic foods. You should stop eating allergenic foods because they irritate the antibodies in the body, and foods that cause allergies vary from person to person, most notably cow's milk, nuts, peanuts, eggs, gluten, fish. 6. Some types of medicine. Some people are allergic to the compounds of some drugs, such as aspirin and penicillin, which causes an increase in the proportion of antibodies and the emergence of various symptoms. Just because your immunoglobulin level is high or low doesn't mean you have one of these conditions. Each person's test can differ based on the method the lab uses to check the results. Talk to your doctor about your test results, and find out what you should do next. Symptoms of increased immunoglobulins. Effect of high levels of antibodies on the body. Despite their effective role in fighting enemies that invade the body, antibodies can have negative effects if their levels are high, such as body allergy, If the appearance of antibodies is caused by an allergy to food, dust, etc., it can cause some symptoms, such as rashes that appear all over the body, shortness of breath. Some types of antibodies also cause difficulty breathing when their levels are high in the body. Sore throat. Elevated types of antibodies can cause a sore throat, especially the types found in the respiratory passages. Abdominal pain. One of the common symptoms of high levels of antibodies in the body is abdominal pain, digestive disorders, and a feeling of severe cramping. The best foods for boosting your immune system. Incorporating specific foods into the diet may strengthen a person's immune response. A healthful, balanced diet plays a vital role in staying well. The following foods may help to boost the immune system. 1. Blueberries. Blueberries contain a type of flavonoid called anthocyanin, which has antioxidant properties that can help boost a person's immune system. A 2016 study trusted source noted that flavonoids play an essential role in the respiratory tract's immune defense system. Researchers found that people who ate foods rich in flavonoids were less likely to get an upper respiratory tract infection, or common cold, than those who did not. 2. Dark chocolate. Dark chocolate contains an antioxidant called theobromine trusted source, which may help to boost the immune system by protecting the body's cells from free radicals. Free radicals are molecules that the body produces when it breaks down food or comes into contact with pollutants. Free radicals can damage the body's cells and may contribute to disease. Despite its potential benefits, dark chocolate is high in calories and saturated fat, so it is important to eat it in moderation. 3. Turmeric. Turmeric is a yellow spice that many people use in cooking. It is also present in some alternative medicines. Consuming turmeric may improve a person's immune response. This is due to the qualities of curcumin, a compound in turmeric. According to a 2017 reviewed trusted source, curcumin has antioxidant and anti-inflammatory effects. 4. Oily fish. Salmon, tuna, pilchards, and other oily fish are a rich source of omega-3 fatty acids. According to a 2014 report, long-term intake of omega-3 fatty acids may reduce the risk of rheumatoid arthritis, RA. RA is a chronic autoimmune condition that occurs when the immune system mistakenly attacks a healthy part of the body. 5. Broccoli. Broccoli is another source of vitamin C. It also contains potent antioxidants, such as sulforaphane. For these reasons, it is a good choice of vegetable to eat regularly to support immune system health. 6. Sweet potatoes. Sweet potatoes are rich in beta-carotene trusted source, a type of antioxidant that gives the skin of the potatoes its orange color. Beta-carotene is a source of vitamin A. It helps to make skin healthy and may even provide some protection against skin damage trusted source from ultraviolet, UV, rays. 7. Spinach. Spinach may boost the immune system, as it contains many essential nutrients and antioxidants, including flavonoids, carotenoids, vitamin C and vitamin E. Vitamins C and E can help support the immune system. Research also indicates that flavonoids may help to prevent the common cold in otherwise healthy people. 8. Ginger. People use ginger in a variety of dishes and desserts, as well as in teas. According to a review, ginger has anti-inflammatory and antioxidative properties and is likely to offer health benefits. However, more research is necessary to confirm whether or not it can effectively prevent illness. 9. Garlic. Garlic is a common home remedy for the prevention of colds and other illness. One trusted source looked at whether taking garlic supplements containing allicin reduced the risk of getting a cold. The group of participants taking a placebo had more than double the number of colds between them than those taking the garlic supplements. However, the researchers concluded that more research is necessary to determine whether or not garlic can help to prevent colds. 10. Green tea. Green tea contains only a small amount of caffeine, so people can enjoy it as an alternative to black tea or coffee. Drinking it may also strengthen the immune system. As with blueberries, green tea contains flavonoids, which may reduce the risk of a cold. 11. Kefir. Kefir is a fermented drink that contains live cultures of bacteria that are beneficial for health. Initial research suggests that drinking kefir may boost the immune system. According to a 2017 review, various studies have shown that regular consumption of kefir can help with fighting bacteria, reducing inflammation and increasing antioxidant activity. The majority of the research that supports this was carried out on animals or in a laboratory. Researchers need to perform additional studies to understand how kefir may prevent disease in humans. 12. Sunflower seeds. Sunflower seeds can make a tasty addition to salads or breakfast bowls. They are a rich source of vitamin E, an antioxidant. In the same way as other antioxidants, vitamin E improves immune function. It does this by fighting off free radicals, which can damage cells. 13. Almonds. Almonds are another excellent source of vitamin E. They also contain manganese, magnesium, and fiber. A small handful or a quarter of a cup of almonds is a healthful snack that may benefit the immune system. 14. Oranges or kiwi fruit, kiwis. Oranges and kiwis are an excellent source of vitamin C, which is the vitamin that many people turn to when they feel a cold developing. While scientists are still not sure exactly how it helps, vitamin C may reduce the duration of common cold symptoms and improve the function of the human immune system. 15. Red bell pepper. For people trying to avoid the sugar in fruit, red bell peppers are an excellent alternative source of vitamin C. Stir-frying and roasting both preserve the nutrient content of red bell peppers better than steaming or boiling, according to a study on cooking methods. The following lifestyle strategies may make a person's immune system stronger. Avoiding smoking, exercising regularly, maintaining a healthy weight, avoiding alcohol or drinking in moderation, getting enough sleep, minimizing stress, practicing correct hand washing and oral hygiene. In the next few minutes we will talk about the antibodies, immunoglobulins, through medical point of view. Therefore, if you are a doctor or pharmacist, this content will be of a great value to you. Antibodies summary. Antibody, molecule in the immune system that circulates in blood and lymph in response to invasion by an antigen. Antibodies are globulins formed in lymphoid tissues by B cells, whose receptors are specialized to bind to a specific antigen. These receptors are copied as antibodies that attack the target antigens by binding to them, either neutralizing them or triggering a complement reaction. Antibodies have widely varying binding sites, providing protection from a wide range of infectious agents and toxic substances. Antibodies derived from the blood serum of infected people or animals are often given in an anti-serum for quick immunization against fast-acting toxins or microbes. In 1975, Cesar Milstein and colleagues developed a process for producing specific antibodies in virtually limitless amounts. These monoclonal antibodies can deliver radiation or drugs directly to specific antigens. Antibodies in medicine and research. Antibodies have important applications in medicine and research. Preformed antibodies, for example, which are derived from the blood serum of previously infected people or animals, are often administered in an anti-serum to another person in order to provide immediate passive immunization against fast-acting toxins or microbes, such as those in snake bites or tetanus infections. Vaccines confer active immunity against a specific harmful agent by stimulating the immune system to attack the agent. Once stimulated by a vaccine, antibody-producing B cells remain sensitized and ready to respond to the agent should it ever gain entry to the body. Monoclonal antibodies, which are produced artificially through genetic engineering and related techniques, are especially valuable in research and medicine. Owing to their ability to recognize individual antigenic sites on almost any molecule, from drugs and hormones to microbial antigens and cell receptors. Immunoglobulins and antibodies. Antibodies, proteins that combat foreign substances in the body, are associated with the globulin fraction of the immune serum. When the serum globulins are separated into alpha, beta, and gamma fractions, antibodies are associated with the gamma globulins. Antibodies can be purified by precipitation with the antigen, i.e., the foreign substance, that caused their formation, followed by separation of the antigen-antibody complex. Antibodies prepared in this way consist of a mixture of many similar antibody molecules, which differ in molecular weight, amino acid composition, and other properties. The same differences are found in the gamma globulins of normal blood serums. The gamma globulin of normal blood serum is thought to consist of a mixture of hundreds of different gamma globulins, each of which occurs in amounts too small for isolation. Because the physical and chemical properties of normal gamma globulins are the same as those of antibodies, the gamma globulins are frequently called immunoglobulins. They may be considered to be antibodies against unknown antigens. If solutions of gamma globulin are resolved by gel filtration through dextrin, the first fraction has a molecular weight of 900,000. This fraction is called IgM or gamma M. Ig is an abbreviation for immunoglobulin and M for macroglobulin. The next two fractions are IgA, gamma A, and IgG, gamma G. With molecular weights of about 150,000 and 320,000 respectively. Two other immunoglobulins, known as IgD and IgE, have also been detected in much smaller amounts in some immune sera. The bulk of the immunoglobulins is found in the IgG fraction, which also contains most of the antibodies. The IgM molecules are apparently pentamers, aggregates of 5 of the IgG molecules. Electron microscopy shows their 5 subunits to be linked to each other by disulfide bonds in the form of a pentagon. The IgA molecules are found principally in milk and in secretions of the intestinal mucosa. Some of them contain, in addition to a dimer of IgG, a secretory piece that enables the passage of IgA molecules between tissue and fluid. The structure of the secretory piece is not yet known. The IgM and IgA immunoglobulins and antibodies contain 10 to 15% carbohydrate. The carbohydrate content of the IgG molecules is 2 to 3%. IgG molecules treated with the enzyme papain split into 3 fragments of almost identical molecular weight of 50,000. 2 of these, called Fab fragments, are identical. The third is abbreviated Fc. Reduction to sulfhydryl groups of some of the disulfide bonds of IgG results in the formation of 2 heavy, or H, chains, molecular weight 55,000, and 2 light, or L, chains, molecular weight 22,000. They are linked by disulfide bonds in the order LHHL. Each H chain contains 4 intricon disulfide bonds, and each L chain contains 2. Antibody preparations of the IgG type, even after removal of IgM and IgA antibodies, are heterogeneous. The H and L chains consist of a large number of different L chains and a variety of H chains. Pure IgG, IgM, and IgA immunoglobulins, however, occur in the blood serum of patients suffering from myelomas, which are malignant tumors of the bone marrow. The tumors produce either an IgG, an IgM, or an IgA protein, but rarely more than one class. A protein called the Bence-Jones protein, which is found in the urine of patients suffering from myeloma tumors, is identical with the L chains of the myeloma protein. Each patient has a different Bence-Jones protein. No 2 of the more than 100 Bence-Jones proteins that have been analyzed thus far are identical. It is thought that one lymphoid cell among hundreds of thousands becomes malignant and multiplies rapidly, forming the mass of a myeloma tumor that produces 1 gamma globulin. Analyses of the Bence-Jones proteins have revealed that the L chains of humans and other mammals are of 2 quite different types, kappa, kappa, and lambda, lambda. Both consist of approximately 220 amino acids. The N-terminal halves of kappa and lambda chains are variable, differing in each Bence-Jones protein. The C-terminal halves of these same L chains have a constant amino acid sequence of either the kappa or the lambda type. The fact that one half of a peptide chain is variable and the other half invariant is contradictory to the view that the amino acid sequence of each peptide chain is determined by one gene. Evidently, 2 genes, one of them variable, the other invariant, fuse to form the gene for the single peptide chain of the L chains. Whereas the normal human L chains are always mixtures of the kappa and lambda types, the H chains of IgG, IgM, and IgA are different. They have been designated as gamma, mu, and alpha chains respectively. The N-terminal quarter of the H chains has a variable amino acid sequence. The C-terminal 3/4 of the H chains have a constant amino acid sequence. Some of the amino acid sequences in the L and H chains are transmitted from generation to generation. As a result, the constant portion of the human L chains of the kappa type has in position 191 either valine or leucine. They correspond to 2 alleles, character-determining portions, of a gene. The 2 types are called allotypes. The valine-containing genetic type has been designated as INV, A+, the leucine-containing type as INV, B+. Many more allotypes, called GM allotypes, have been found in the gamma chains of the human IgG immunoglobulins. More than 20 gigameters allotypes are known. Certain combinations of GM types occur. For example, the combination of GM types 5, 6, and 11 has been found in Caucasians and African Americans but not in Chinese. The combination of 1, 2, and 17 has not been found in African Americans, and the combination of 1, 4, and 17 has not been found in Caucasians. Allotypes have also been discovered to occur in a number of other animals, including rabbits and mice. Activation of killer cells. Some cells that bear antigen-antibody complexes do not attract complement. Their antibody molecules are far apart on the cell surface or are of a class that does not readily activate the complement system, example, IgA, IgD, and IgE. Other cells have outer membranes that are so tough or can be repaired so quickly that the cells are impermeable to activated complement. Still others are so large that phagocytes cannot ingest them. Such cells, however, can be attacked by killer cells present in the blood and lymphoid tissues. Killer cells, which may be either cytotoxic T cells or natural killer cells, have receptors that bind to the tail portion of the IgG antibody molecule, the part that does not bind to antigen. Once bound, killer cells insert a protein called perforin into the target cell, causing it to swell and burst. Killer cells do not harm bacteria, but they play a role in destroying body cells infected by viruses and some parasites. Other antibody-mediated mechanisms. The protection conferred by IgA antibodies, which are transported to the surface of mucous membrane-lined passages, is somewhat different. Complement activation is not involved. There are no complement proteins in the lining of the gut or the respiratory tract. Here the available immune defense mechanism is primarily the action of IgA combining with microbes to prevent them from entering the cells of the lining. The bound microbes are then swept out of the body. IgA also appears to direct certain types of cell-mediated killing. IgE antibodies also invoke unique mechanisms. As stated earlier, most IgE molecules are bound to special receptors on mast cells and basophils. When antigens bind to IgE antibodies on these cells, the interaction does not cause ingestion of the antigens but rather triggers the release of pharmacologically active chemical contents of the cell's granules. The chemicals released cause a sudden increase in permeability of the local blood vessels, the adhesion and activation of platelets, blood cell fragments that trigger clotting, which release their own active agents, the contraction of smooth muscle in the gut or in the respiratory tubes, and the secretion of fluids, all of which tend to dislodge large multicellular parasites such as hookworms. Eosinophil granulocytes and IgE together are particularly effective at destroying parasites such as the flatworms that cause schistosomiasis. The eosinophils plaster themselves to the worms bound to IgE and release chemicals from their granules that break down the parasite's tough protective skin. Therefore, IgE antibodies, although they can be a nuisance when they react with otherwise harmless antigens, appear to have a special protective role against the larger parasites. Transfer of antibodies from mother to offspring. A newborn mammal has no opportunity to develop protective antibodies on its own, unless, as happens very rarely, it was infected while in the uterus. Yet it is born into an environment similar to its mother's, which contains all the potential microbial invaders to which she is exposed. Although the fetus possesses the components of innate immunity, it has few or none of its mother's lymphocytes. The placenta generally prevents the maternal lymphocytes from crossing into the uterus, where they would recognize the fetal tissues as foreign antigens and cause a reaction similar to the rejection of an incompatible organ transplant. What is transferred across the placenta in many species is a fair sample of the mother's antibodies. How this happens depends on the structure of the placenta, which varies among species. In humans, maternal IgG antibodies, but not those of the other immunoglobulin classes, are transported across the placenta into the fetal bloodstream throughout the second two-thirds of pregnancy. In many rodents a similar transfer occurs, but primarily across the yolk sac. In horses and cattle, which have more layers of cells in their placentas, no antibodies are transferred during fetal life. and the newborn arrives into the world with no components of specific immunity. There is, however, a second mechanism that makes up for this deficiency. The early milk, colostrum, is very rich in antibodies, mainly IgA but also some IgM and IgG, and during the first few days of life the newborn mammal can absorb these proteins intact from the digestive tract directly into the bloodstream. Drinking colostrum is therefore essential for newborn horses and cattle and required to a somewhat lesser extent by other mammals. The capacity of the digestive tract to absorb intact proteins must not last beyond 1 or 2 weeks, since once foods other than milk are ingested, the proteins and other antigens in them would also be absorbed intact and could act as immunogens to which the growing animal would become allergic. IgA in milk is, however, rather resistant to digestion and can function within the gut even after intact absorption into the bloodstream has ended. Human colostrum is also rich in IgA, with the concentration highest immediately after birth. After a newborn has received its supply of maternal antibodies, it is as fully protected as its mother. This means, of course, that if the mother has not developed immunity to a particular pathogen, the newborn will likewise be unprotected. For this reason, a physician may recommend that a prospective mother receive immunizations against tetanus and certain other disorders. The active immunization of pregnant women against certain viral diseases, such as rubella, German measles, must be avoided, however. Because the immunizing agent can cross the placenta and produce severe fetal complications. As important as the passively transferred maternal antibodies are, their effects are only temporary. The maternal antibodies in the blood become diluted as the animal grows. Moreover, they gradually succumb to normal metabolic breakdown. Because the active development of acquired immunity is a slow and gradual process, young mammals actually become more susceptible to infection during their early stages of growth than they are immediately after birth. Occasionally the transfer of maternal antibodies during fetal life can have harmful consequences. A well-known example of this is erythroblastosis fetalis, or hemolytic disease of the newborn, a disorder in which maternal antibodies destroy the child's red blood cells during late pregnancy and shortly after birth. The most severe form of erythroblastosis fetalis is Rh hemolytic disease, which develops when 1. The fetus is Rh positive, that is, its red blood cells carry an antigen known as the Rh factor. 2. The mother is Rh negative, which is to say her red blood cells lack the Rh factor. 3. The mother's immune system has been previously activated against the Rh antigen. This usually is the result of exposure to fetal cells during the birth of an earlier Rh-positive baby or a transfusion of Rh-positive blood. Rh hemolytic disease can be prevented by giving the mother injections of anti-Rh antibody shortly after the birth of an Rh-positive child. This antibody destroys any Rh-positive fetal cells in the maternal circulation, thereby preventing the activation of the mother's immune system should she conceive another Rh-positive fetus. Cell-mediated immune mechanisms. In addition to their importance in cooperating with B cells that secrete specific antibodies, T cells have important separate roles in protecting against antigens that have escaped or bypassed antibody defenses. Immunologists have long recognized that antibodies do not necessarily protect against viral infections. Because many viruses can spread directly from cell to cell and thus avoid encountering antibodies in the bloodstream. It is also known that persons who fail to make antibodies are very susceptible to bacterial infections but are not unduly liable to viral infections. Protection in these cases results from cell-mediated immunity, which destroys and disposes of body cells in which viruses or other intracellular parasites, such as the bacteria that cause tuberculosis and leprosy, are actively growing, thus depriving microorganisms of their place to grow and exposing them to antibodies. As discussed in the section Activation of T and B lymphocytes, cell-mediated immunity has two mechanisms. One involves activated helper T cells, which release cytokines. In particular, the gamma interferon produced by helper T cells greatly increases the ability of macrophages to kill ingested microbes. This can tip the balance against microbes that otherwise resist killing. Gamma interferon also stimulates natural killer cells. The second mechanism of cell-mediated immunity involves cytotoxic T cells. They attach themselves by their receptors to target cells whose surface expresses appropriate antigens, notably ones made by developing viruses, and damage the infected cells enough to kill them. Cytotoxic T cells may kill infected cells in a number of ways. The mechanism of killing used by a given cytotoxic T cell depends mainly on a number of co-stimulatory signals. In short, cytotoxic T cells can kill their target cells either through the use of pore-forming molecules, such as perforins and various components of cytoplasmic granules, or by triggering a series of events with the target cell that activate a cell death program, a process called apoptosis. In general, the granular cytotoxic T cells tend to kill cells directly by releasing the potent contents of their cytotoxic granules at the site of cell-to-cell contact. This renders the cell membrane of the target cell permeable, which allows the cellular contents to leak out and the cell to die. The non-granular cytotoxic T cells often kill cells by inducing apoptosis, usually through the activation of a cell surface protein called FAS. When a protein on the surface of the cytotoxic T cell interacts with the FAS protein on the target cell, FAS is activated and sends a signal to the nucleus of the target cell, thus initiating the cell death process. The target cell essentially commits suicide, thereby destroying the virus within the cell as well. Immunity against cancer. Cancer cells are normal body cells that have been altered in a manner that allows them to divide relentlessly, ignoring normal signals of restraint. As a result, cancer cells form clusters of cells, called tumors, that invade and colonize tissues, eventually undermining organ function and causing death. In the early 20th century, the pioneering immunologist Paul Ehrlich pointed out that the enormous multiplication and differentiation of cells during prenatal life must afford many opportunities for aberrant cells to appear and grow, but that immune mechanisms eliminate such cells. The idea that such a mechanism continues to function throughout life, weeding out newly arisen cancer cells, became popular in the 1950s and 60s when a number of immunologists postulated immune surveillance. The theory that T-cell-mediated immunity evolved as a specific defense against cancer cells and that T cells constantly patrol the body, searching for abnormal body cells that carry antigens on their surface which are not found on healthy body cells. Although it has its compelling aspects, the immunosurveillance theory remains just a theory, and a controversial one at that. The role of the immune system in protecting against cancer has not been fully explained. But nevertheless, there is no question that in some instances the immune system can distinguish cancer cells from normal cells. The study of tumor immunology has shown unequivocally that cancer cells do carry antigens that are not present on healthy cells. Immunologists distinguish broadly between two types of tumor antigens: tumor-specific antigens, which are found only on cancer cells and not on their normal counterparts, and tumor-associated antigens, which are found on both normal and cancer cells but which are abnormally expressed, for example, are overproduced, on cancer cells. In both cases, these antigens have been shown to evoke an immune response, although not necessarily one strong enough to eliminate the tumor. Why does a tumor continue to grow if an immune response against it is induced? Through animal experiments. A number of mechanisms have been identified that allow tumors to avoid recognition and destruction by the immune system. 1. The surfaces of cancer cells may lose antigens that are recognizable by the immune system. 2. Cancer cells may lose all class I MHC molecules from their surface, which prevents cytotoxic T cells from recognizing the cells. 3. Some cancer cells produce immunosuppressive chemicals that can inhibit T cells directly or that can activate regulatory T cells. 4. Some cancer cells shed some of their antigens, and these newly released, free-floating antigens may bind to the receptors on cytotoxic T cells, plugging them up so that the T cells cannot bind to the cancer cells and eliminate them. 5. Certain cancer cells can outmaneuver an immune response by growing so rapidly or becoming such a dense mass that immune cells cannot come in contact with most of them. Other dysfunctions of the immune system, such as immune suppression and immune deficiency, may contribute to cancer development and growth. Individuals such as transplant patients who have been treated with immunosuppressive drugs for a long period of time are more likely to develop certain types of cancer, as are patients with immunodeficiency diseases. For example, people with AIDS, acquired immunodeficiency syndrome, are more prone to developing cancers associated with viruses, such as Kaposi sarcoma. The incidence of cancer also increases greatly in old age, when some immune responses decline. But defective immune responses may not be the major factor involved in cancer development in the elderly, since genetic mutations that are linked to cancer also accumulate with age. Much research has been devoted to developing effective immunotherapies against cancer. One avenue of research has focused on finding ways to immunize patients against the specific cancer growing within them. This approach targets tumor-specific antigens found on the cancer cells. Because these antigens are altered forms of normal self-antigens, they are foreign, and could be recognized by the immune system as such, but often they are not. However, investigators are working to develop vaccines that stimulate an immune response to these antigens, hoping that the reaction would be strong enough to eliminate the cancer. Advances in scientists' understanding of mechanisms underlying T-cell activation and immune regulation have facilitated the development of human monoclonal antibodies specifically tailored for the treatment of certain cancers. For example, the realization that a molecule known as CTLA-4 plays a role in downregulating immune responses against tumors led to the development of a therapeutic human monoclonal antibody against CTLA-4. Known as ipilimumab, Yervoy, the antibody was approved in 2011 by the U.S. Food and Drug Administration for the treatment of late-stage melanoma. Likewise, the discovery of a negative immune regulatory protein known as programmed cell death protein 1, PD-1, which occurs on the surface of T cells, led to the development of highly effective anti-PD-1 cancer immunotherapies. Prophylactic immunization. Prophylactic immunization refers to the artificial establishment of specific immunity, a technique that has significantly reduced suffering and death from a variety of infectious diseases. There are two types of prophylactic immunization: passive immunization, in which protection is conferred by introducing preformed antibodies or lymphocytes from another individual whose immune system was stimulated by the appropriate antigen. Active immunization, in which protection results from the administration of a vaccine, with dead or harmless living forms of an organism or with an inactivated toxin, that stimulates the immune system to produce lymphocytes and antibodies against that organism or toxin. Passive immunization. It is sometimes the case that an infectious organism or a poisonous substance can have such a rapid deleterious effect that the victim does not have time to develop an immune response spontaneously. At such times, passive immunization with preformed antibodies can provide life-saving assistance in combating the pathogen or poison. This situation may arise in victims of poisonous snake bites or botulism, as well as in those in whom such infections as diphtheria, tetanus, or gas gangrene have progressed to the point at which bacterial toxins have been absorbed into the bloodstream. It is also the case with bites from a rabid animal. Although active immunization is begun at the same time, since the spread of the rabies infection to the central nervous system is relatively slow, physicians use passive immunization as temporary protection for persons traveling to countries where hepatitis B is prevalent. Passive immunization provides antibodies to persons who suffer from B-cell deficiencies and are therefore unable to make antibodies for themselves. See immune system disorder, immune deficiencies. Also, as discussed earlier, passive immunizations of anti-Rh antibody can prevent erythroblastosis fetalis. Protective immunoglobulins, primarily of the IgG class, can be prepared from the blood of humans or other species, for example, horses or rabbits, that have already developed specific immunity against the relevant antigens. These preparations are known as antiserums. This explains the original term for passive immunization, which is serum therapy. Human IgG is slowly broken down in the recipient's body, the concentration falling by about 1/2 every 3 weeks, so that effective amounts of antibody can be present for 2 or 3 months. Human antiserum is used whenever it is available. Because IgG from other species is far more likely to provoke an immune response that will eliminate the antibody and may lead to serum sickness. Active immunization. Active immunization aims to ensure that a sufficient supply of antibodies or T and B cells that react against a potential infectious agent or toxin are present in the body before infection occurs or the toxin is encountered. Once it has been primed, the immune system either can prevent the pathogen from establishing itself or can rapidly mobilize the various protective mechanisms described above to abort the infection or toxin in its earliest stages. The vaccines used to provide active immunization need not contain living microbes. What matters is that they include the antigens important in evoking a protective response and that those antigens be administered in a harmless form sufficient in amount and persistence to produce an immune response similar to the natural infection. Bacterial toxins, such as those that cause tetanus or diphtheria, can be rendered harmless by treatment with formaldehyde without affecting their ability to act as immunogens. These modified toxins, or toxoids, usually are adsorbed onto an inorganic gel before being administered, an approach that increases the likelihood that the toxoid will be retained in a macrophage. Toxoids elicit effective, long-lasting immunity against bacterial toxins. When immunization against several antigenic determinants is desired or the important antigenic component is not known. It may be prudent to use the entire microbe, which has been killed in a manner that does not alter it significantly. Such so-called killed vaccines are used to immunize against typhoid, pertussis, whooping cough, plague, and influenza, for example. In other cases, researchers have developed attenuated, i.e., weakened, strains of bacteria or viruses. Attenuated vaccines cause an infection but do not produce the full array of signs and symptoms of the disease. Because the infectious agent multiplies to only a limited extent in the body and never reverts to the virulent form. The use of such live microbes provides the most effective prophylaxis of all, since they truly imitate a mild form of the natural infection. Such are the vaccines for yellow fever, poliomyelitis, oral vaccine, measles, rubella, and tuberculosis. Although sufficiently attenuated as far as healthy persons are concerned, live vaccines may cause the full disease in persons who have an immune deficiency. Most vaccines are administered by injection, but a few are given orally. Ultimately, mucosal vaccines, those administered to mucosal surfaces such as those lining the gut, nasal passages, or the urogenital tract, may be the most effective vaccines available because of their unique ability to stimulate IgA responses and because of their ease of administration. Recombinant DNA technology has allowed researchers to use modified bacteria and viruses that are not harmful to humans to immunize individuals against an antigen from a pathogenic microorganism. This approach involves introducing into the DNA of the harmless microorganism a gene from a pathogenic organism that encodes an antigen capable of eliciting a protective immune response but not their full-blown disease. Once inoculated into the host, the microorganism generates the protective antigen of the pathogen and immunizes the host. An effective oral vaccine against cholera was developed based on this approach. Sometimes different strains of a microorganism, each characterized by a different antigenic determinant, give rise to the same disease. In such cases, neither natural infection nor prophylactic immunization with any one strain protects against infection by the others. For example, a variety of virus strains cause the common cold. But it is impractical to immunize against each strain. On the other hand, although there are more than 60 different strains of pneumococci that can cause bacterial pneumonia, some strains are much more common than others. Consequently, a vaccine containing antigens from up to 14 of the most common strains is useful in protecting persons at special risk. Active immunization is often the most effective and least costly method of protecting against an infectious disease. Vaccination campaigns against many diseases, such as diphtheria, polio, and measles, have been tremendously successful. In cases in which 95% or more of the population at risk is protected and humans are the only reservoir of infection. Active immunization can lead to the worldwide eradication of the infectious agent, as has been achieved in the case of smallpox. Genetic origins of the immune system. Researchers have found many similarities between the structures of proteins involved in antigen recognition and those in cell-to-cell recognition in the immune system. Antigens are the foreign proteins that antibodies recognize and bind to. These proteins include the antigen receptors of lymphocytes, the MHC proteins. The coreceptors involved in cell-to-cell recognition in immune reactions, such as the receptors named CD4, CD8, and CD28. And the Fc receptor that binds to the stem of the Y-shaped immunoglobulin molecule. A number of proteins not involved in the immune system also share structural features with these proteins. The main feature similarity is a structure called the immunoglobulin domain. Each protein is composed of one or more Ig domains of nearly identical size. The domains are formed into a loop by bonds between sulfur atoms on the amino acids at the ends. Although each domain is different and serves a different function in the molecule as a whole, the number and order of the amino acids forming each domain are far more similar than would be expected if each had arisen independently in the course of evolution. Equally remarkable is the fact that nerve cells, thymus cells, and T lymphocytes, T cells, in mice and rats carry a surface protein called Thy1, thymus 1 antigen, that also has this same basic structure and a similar arrangement of amino acids. The similarities suggest that the genes for all these molecules originated from some primitive gene involved in the recognition of one cell by another, which is required for orderly development of a complex, multicellular organism, and that during evolution they had acquired different functions. Researchers named this group of genes and their protein products the immunoglobulin superfamily. The processes whereby one ancestral gene could have given rise to such a family of genes include gene duplication, crossing over, and mutation, all of which are discussed in detail in genetics. Not surprisingly, molecules that have a similar function in different species, for example, immunoglobulins or MHC components, show an even closer resemblance. By analyzing the number of differences in the amino acids and their position in the polypeptide chains that make up IgM and IgG molecules in, for example, humans, mice, and rabbits, and by making reasonable assumptions about mutation rates, scientists can estimate roughly how many generations would have had to elapse, and hence how much time, for the present immunoglobulins of these species to have evolved from a common ancestral IgM-like molecule. Such calculations suggest that divergence from the ancestral immunoglobulin took place some 200 million years ago. That was about the same time amphibians are thought to have diverged from the main vertebrate line. So one may conclude that a functional immune system arose even earlier and has continued to provide a defense against foreign agents ever since. We wish you health and wellness. If you like this video, do not forget to subscribe to the channel, press the like button and activate the bell to receive all new